Hello and welcome to this presentation on inferring time-delayed gene regulatory networks using sparse regression and cross-correlation. I am Piyushkumar Mundra from School of Computer Engineering, NTU Singapore, and this work is done in collaboration with Professor Mahesan Niranjan from University of Southampton and Professor Roy Welsch from MIT and our group in NTU. With microarrays, as we know, it is possible to measure the expression of thousands of genes simultaneously. However, the data are only gathered over a few time samples for several reasons, such as cost of experiments, availability of samples, etc. So, using such data, biologists are generally interested in identifying the underlying biology, which could be generally represented by gene regulatory networks. And in past decade, several of techniques are available for building GRNs. These include Bayesian networks, linear regression framework, mutual information-based approach, and many, many more. However, in many of the previously developed methods to model a GRN, it is generally assumed that gene expression at a given time point only depend on the immediate previous time point. I'm particularly talking about the time series data and the GRN inference using time series data. So such an assumption leads to GRN with first order parenting, or also known as the first order delay or lag. In reality, however, in most of the cases, the regulation of one gene by another gene may occur only after a certain number of time points, which actually results in invalid first order assumption. To identify such higher order delays, novel approaches are needed, as the model is need to work with less number of available time points, as well as there will be a significant increase in the parameter space. So many models which predict significant number of false positives, those are because of these reasons of less number of time points and increased parameter space. So in this paper, we propose a simple yet very effective solution to model a higher order GRN under a sparse linear regression framework. This is basically a two-step method. So we first determine probable order of regulations using cross correlation in the first step, and in the second step, lasso, which is actually a least absolute shrinkage and selection operator based regression in a multivariate autoregression framework to infer a time delayed GRN. So before I go to the details of the proposed approach, let me first explain a general linear regression based framework to identify regulations from long time series data. So let capital X equals to xit, small xit, denotes the expressions of I genes gathered over equally spaced t time points. A higher order fully connected network of these I number of genes could be derived by using a rth order multivariate autoregressive model, as shown in this equation. Xt equal to summation of tau equal to 1 to r beta tau x t minus tau plus epsilon t. So this is very popular equation in signal processing, and in this equation, beta tau represents the strength of the interactions, and this is also a regression coefficient between all the pairs of genes for a model of order tau, and epsilon t denotes the residuals that are assumed to follow a Gaussian distribution with zero mean and are independently and identically distributed. And as you see, the R here denotes the maximum number of time delay. And from this equation, it's also very clear that if there is a rth order model, we'll need to estimate I square R coefficients. So in terms of gene expression data, if we have I genes, which is generally in thousands, so the I square number of parameters multiplied by R, I square multiplied by R coefficients will need to estimate. The MBR model, which was discussed in the last slide, it needs to be modified for inference related to biological networks, such as time-delayed gene regulatory networks, essentially for these three reasons. First of all, it is generally assumed that expression time series data are stationary, and there cannot be multiple regulation ages with different time legs exist between two particular genes. Second, biologically, it is very well known fact that GRNs are sparse in nature, while in a standard formulation, which was shown on the previous slide, that will give us a fully connected network. And last but not the least, from a computational point of view, in a typical time series gene expression data, the number of genes whose expressions are measured are far higher than number of time samples. Hence, a very simple linear standard regression technique to derive strengths of connection is inapplicable. So the question is how to tackle these three challenges. So in this paper, we develop an algorithm to tackle these challenges by first using cross correlation, and in the second step using a sparse regression techniques such as lasso, which will finally give us a time delayed network. So this is the first step where we fix the delays using cross correlation. So if you think mathematically, the assumption of a single time delayed regulation out of all possible R legs between say two genes I and J, this implies that for any tau value, if beta tau ij between these two genes I and J is greater than 0, then the rest of the beta coefficient with other tau values for these two genes I and J, that is the J regulating I gene, should be 0. And this requirement could be achieved by using the cross correlation between two genes and using the legs that gives maximum absolute cross correlation. For example, if gene J regulates gene I, the unbiased cross correlation is given by the equation shown on the slide. And here C hat is an estimated unbiased cross correlation for regulation of gene I by gene J, and such cross correlation was computed after normalizing expressions of a gene to have zero mean and a standard deviation of 1. And these values are computed for all tau values, that is tau equal to 1, 2, 3 to R, and the maximum of the absolute cross correlation denotes the probable possible time lag regulation. So once the probable time legs are fixed using the cross correlation as shown on the previous slide, the next step is to identify the relevant regulators for gene I from all the possible genes, and this can be done by employing sparse linear regression techniques such as lasso. So continuing with our assumption that gene J regulates gene I with the time lag of Kij, where Kij is the estimated time lag using cross correlation, and let's denote yt and zt as shown on the second line of this slide, and note the time samples in yt and zt, which are r plus 1 to t and r minus kij plus 1 to t minus kij. So these represents the adjustment from the time delayed information, and then using a multivariate vector autoregressive model, we can very simply model yt equal to zt into beta star plus epsilon t, and of course the beta star is the regulation strength, and this will be the matrix of size I times I, which is number of genes into number of genes, and epsilon t will denote the corresponding errors or the innovations. So if we assume if we assume y and zt in a capital y and zt, those are the corresponding matrix form of small yt and small zt, then the beta star coefficient values can be very easily estimated using a very simple linear regression based technique, and the solution is very straightforward as shown on the second equation of this slide. However, considering that GRNs are sparse, and more importantly, the number of time samples are significantly smaller than number of genes in a typical gene expression data, this beta solution is cannot be achieved. And however, we can use lasso regression, and the loss function of this lasso equation is shown on the last equation of this slide. And as you can see, there is a parameter alpha I, which is a regularization parameter, and it forces some of the beta coefficients to be exactly zero, and this gives us a sparse solution in terms of beta regression parameters or regression coefficients. The essence of last two slides is shown in this algorithm, as it describes the complete approach to derive a time-delayed GRN. As discussed, this is basically a two-step procedure. So we start with a time series gene expression data and a fixed maximum time delay, that is R, and an empty final matrix of time delay information, that is K. For a given gene I, cross correlation is used in the first for loop to determine the probable time lags of regulations by other genes. And in the second step, lasso regression is used to derive the regulators. By repeating the same process for all the genes, a complete time-delayed GRN is derived, and both the estimated regression coefficients and associated time delay information is stored. So after describing our method, I now move on to the experimental part. And this slide basically depicts the procedure to generate a synthetic data set, because we test our method on both synthetic as well as on the real data set. So to generate a synthetic data set, we first extract a network using a topology of global yeast network and GNW software. Though this GNW software can also provide data sets very popularly known as Dream Challenge data sets, it does not have features to generate the time-delayed data sets. So we need to devise a simple framework to generate data set which has the time-delayed information. So after extracting a network topology, all the pairs of genes which are not connected, the regression coefficients corresponding to these no interactions among genes were set to 0, and all the available edges were randomly assigned a time-delayed value tau, which is between 1 to r. So for all the edges with respective tau values, regression coefficients were obtained by drawing samples from a uniform distribution on the interval of 0.08 to 1, and coefficients for all other time lags in R were set to 0. For example, if jth gene regulates the ith gene with second order time delay and R is set to 5, then beta ij for the second order will be between 0.8 to 1, and rest all beta values will be equal to 0. Now the initial gene expression values at t equal to 0, 1, 2 to r were drawn from a uniform distribution on the interval 0 to 1, and for successive time points, expressions were generated using a higher order MVR model with added IID Gaussian random noise. And the first 10,000 samples were discarded, and the number of time points were varied from 20, 30, or 40. And for each combination of network size and number of time points, we generated 100 time series datasets by randomly initializing the gene expressions. So to demonstrate the performance on real dataset, we selected the Saccharomyces cerevisiae, that is yeast cell cycle dataset. And this dataset is used in 2 recent studies and is available with the PDRNA package. And this is a very popular dataset to infer gene regulatory networks. And basically, Spellman et al. Have identified 800 differentially expressed genes for cell cycle regulation covering 4 phases, G1, S, G2, and M for yeast development. And for our analysis, we choose 11 genes from CDC28 experiments, and these genes were CLN3, CDC28 to MBP1. And these genes were taking part in G1 phase, and they have 17 time points. However, as suggested in previous literature, the first time point was excluded as it is related to the M step, and hence the final dataset had 11 genes and 16 time points. And generally, the ground truth of the real dataset is not available, so we will use the yeast KEGG pathway to validate our parameter estimation and the way we evaluate our performance, that is the critical part of our algorithm. So in the proposed algorithm, the second step of lasso regression is used to identify regulatory edges and to generate sparse time-delayed GRNs. So the network topology is essentially achieved by I-separate lasso regressions, and the lasso solutions were achieved by using GLMNet package, which can generate the whole solution path alpha I. For each regression, the penalty parameter alpha I was chosen by using 5-fold cross-validation. And another important thing is that in both synthetic as well as in the real dataset, expressions of genes were normalized to have 0 mean and 1 standard deviation even before computing the cross correlation. And for real dataset, to avoid errors due to parameter estimation with single 5-fold cross validation, we repeated the 5-fold cross validation process for 100 times and used edge stability of 0.75 to infer the final single network structure. This basically represents that out of 100 iteration, at least 75 times the same edge was obtained. And the performance of the proposed approach was evaluated over 100 simulated data sets for each combination of number of genes and number of time points. And in generating simulated data set, we had the network topology. So basically we had the ground truth available. That is why we used precision, recall, and F measure as the performance metric. However, here the TP were computed for exact time delays, while FVs were computed for counting all instances where false edge for any time order was detected. And as you can see, we have also defined order identification accuracy, which is OIA. This is the number of edges which were identified with true time delays among all the total number of identified true edges irrespective of time order. Here are the results on synthetic datasets with maximum delay set to 3. As you see, the increase in number of genes and decrease in length of time series reduces precision, recall, as well as F measure. And the results also show that within truly identified edges, the correct delay is also generally identified with a very high accuracy. Here are the results on synthetic dataset with maximum delay set to 5. Again, as you see, the increase in number of genes and decrease in length of time series reduces precision, recall, and F measure. And the result also show that within truly identified edges, the correct delay is generally identified with a very high accuracy. Another interesting fact is that by fixing the lower value of maximum possible time delay, the performance could be improved because a single point increase in maximum delay, that is the value of R, increases the number of parameters to be estimated by square of number of genes. And the available number of time samples actually reduces by 1. So it is important not to choose a very high value of a maximum possible time delay to get any meaningful results by the proposed algorithm. As I discussed earlier, as a true underlying network of yeast is unknown, we will use the KEGG pathway to validate the reconstructed time delayed GRN. So basically in a yeast cell cycle progression, G1 and G2 phases are the gaps between DNA replication S phase and mitosis, which is M phase. So as per KEGG pathway and the previous literature, in G1 phase, an association between CLN3 and CDC28 is needed to initiate the start of the cycle. So after reaching a certain threshold of CLN3 and CDC28 complex, two transcription factor SBF and MBF are activated, and SV4 and SV6 form the SCB complex with SBF, which results in activation of CLN1 and CLN2 genes, while MBP1 and SWI6 form a complex with MBF to promote transcription of other genes required for S-phase progression. CLN1 and CLN2 interacting with CDC28 promote the activation of B-type cyclin-associated CDK, which drives DNA replication and entry into mitosis. Further, CLB1 and CLB2 are associated with CDC28, and this complex represses SIC1, which in turn represses the CLB5, CLB6, and CDC28 complex. So we'll basically use this information to see if the proposed method gives any meaningful interaction results or not. The GRNs inferred by the proposed two-step method with R equal to 3 is shown in this figure. As can be seen, several two gene-gene interactions have been recovered. For example, in this figure, we find interaction between CLN3 and SWI6, CLB6 and CDC6, interaction of SIC1 with CLN1, CLB6 and CLN2, SWI4 and SWI6, and an interaction of SWI4 and SWI6 with CLN1 and CLN2. However, we also note that there are few wrong directions of regulations which we have discovered with our proposed method. This slide gives time-delayed GRN inferred with R equal to 5, and comparing between the previous slide and this figure reveals that few new edges are formed and few are not recovered. And we believe that such phenomena could be attributed to loss of time samples and increase in the parameter space. So as these results have shown, building a time-delayed GRN is a very challenging problem, particularly because if we set a maximum time delay R to a very high value, there will be a significant increase in number of parameters which we will need to estimate, and this will also reduce the number of available time samples which are already very scarce, and this will basically result in significant false positives. So we have a challenge to how to improve the predictions by reducing the number of false predictions. This could be done in several ways. For example, in our previous work, we have developed a bootstrapping technique for short time series dataset with a first order assumption. And we believe that developing such techniques for higher order models and integrating stability criteria will possibly improve the performance. Further, in the current two-step procedure, cross correlation was used to determine the probable time lags. Now we know that cross correlation may suffer due to small sample size, so developing a robust technique with possibly a single step procedure would be another interesting extension of this work. And last but not the least, an extension of the data integration approach such as PPI data or histone enrichment data to higher order models may actually help us in deriving a highly accurate time-delayed GRN. Thank you for your attention, and if you have any questions or suggestions, please contact me at this email ID. Thank you.